Good evening and welcome to Montana Tech's first Café Scientifique of the 2018-2019 academic year. This is also the first week of class, and so we've got really a great turnout tonight. So raise your hand if you're a tech student, any level. Raise your hand if you're a member of the public, not working at Tech or for Tech. And then raise your hand if you're a faculty or staff here. So you see you have a mixed.
All right.
So now raise your hand if I didn't put you in any category at all, but you didn't identify.
No one's going to put their hands
So
up.
identify with no category. Anyway, thank you all for coming tonight. I'm Bev Hartline. I'm the Vice Chancellor for Research and Dean of the Graduate School, and it's a particular pleasure to have managed to convince Dr. Sarjubhai Patel from the University of Montana to visit us this afternoon to talk about something that's been in the news a lot over the last couple of years, and particularly as football season is getting underway, it's going to be in the news some more, and that's traumatic brain injury. Dr. Patel got his PhD, I think.
Yeah.
He got his undergraduate degree in England at the University of Sunderland.
That's right.
And then he earned a Master's of Science in Neuroscience in King's College London, followed by a PhD in Pharmaceutical Sciences right down the road at the University of Montana. He spent a bit of time away at Johns Hopkins University, then he came back to the University of Montana, where he's an associate research professor of molecular pharmacology at the Skaggs School of Pharmacy. Now, if those of you who don't know, if it's called research professor, that means you have to generate all the grant money that's going to pay your salary and the salary of your technicians, graduate students, and colleagues. And so that's an extra challenge compared to the challenge of teaching and also integrating into that some research. He has been extremely successful on this work on TBI. He's landed so far 3 grants. He was one of 16 out of more than 200 who was awarded a pilot grant from the GE National Football League Research Fund for TBI. And then he was one of 5, the 5 who was given a second phase there. But he also was successful in the Montana Research
Academic Development Initiative.
Development Initiative a couple of years ago, where the legislature appropriated $15 million for research across Montana, and he was part of a collaboration at UM, which together got $2.3 million, and all this has happened, I think, within the last 4 years.
4 or 5 years, yeah.
Been going on. So please join me in welcoming Dr. Patel, and I think we're going to have a great talk this afternoon.
I hope so. Thank you.
And I need to say is that these Café Scientifiques are sponsored by the Montana Embry Idea Network for Biomedical Research Excellence, which is funded by the National Institutes of Health, and it's headquartered at the Montana State University. So we need to thank Embry for the food and also for the speaker.
Thank you, Beverly. Thank you for that kind introduction, and thank you all for turning up on an evening to hear this talk. As Beverly said, we in our program have primarily been focusing on studying how traumatic brain injury causes molecular changes in the brain. Okay, for a long period of time, anybody who's taken part in any kind of organized sports, especially where there's contact, there was a history of head injuries being sort of swept under the rug. A lot of the attitude was shake it off, walk it off kind of things, and people were ignoring the signs of some really severe symptomology that was, we believe, directly related to those initial injuries. And so over the period of the next hour, I'm going to give you a little idea of some of the research that we did under the MReady Award, which really helped us kickstart some really interesting avenues of research at UM and further afield across the state of Montana and also around the world through collaborations. And so with that, I'm going to just start by got to make some disclosures. I do have some financial incentives outside of my research. We've got a company that sort of stemmed off from the MReady work that's developing some of the technology that we did at and then we've also licensed some of our technology to a company in Australia that's trying to do some validation work with these biomarkers. So what was the MReady? The MReady was a state initiative run out of MUS to basically take technology that researchers have developed on the campuses of all the universities and institutions of higher learning across the state. And it was sort of an open competition to look at specific areas of research. And we were sort of under the health and medicine umbrella. And we put together a consortium of researchers and a couple of private companies very much focusing on trying to understand and develop new technologies for traumatic brain injury, because at the time we believed that what currently existed, and I'll talk a little bit about that as the talk progresses, they just weren't adequate to address the needs of the individuals who are suffering from these injuries, both in terms of detection of the disease, tracking the disorder, and then also some of the other outcomes, some of the post-traumatic injuries and symptomologies that can arise. And so there was quite a large breadth of projects that were sort of covered under the grant. One of the big things that was an important part that really helped set up the Neural Injury Center at the University of Montana was basically we have close to 500 veterans, student veterans on campus, and a large portion of those have seen active time in the arena of war, and had a major portion of those had suffered traumatic brain injuries. And part of the NIC was to help those individuals navigate getting through college, because one of the big factors in reaching graduation is about more than half of those students, after they enroll their freshman year, don't go on to graduate. So we lose almost more than half of those students. So that was a major part, providing support for those particular students. And it sort of stemmed off from that was a series of projects where we could take some of the research we were doing at the university and see how we could help those particular students. And very generally, a lot of these things were diagnostics based, all right? So we were trying to find ways to track TBI and see how one, the brain was reacting, and what outcomes we could use as measures of those injuries. And one of the things that I'm going to talk mostly about today is this number 2, where we were developing a set of biological tests that we could use to better understand, one, what's happening in the brain, get an idea of the underlying mechanisms that occur after the injury, many months and years after the injury, as well as giving us an idea of if the brain is healing, if it's continuing to deteriorate, using various methodologies, blood-based testing, oculomotor, so basically looking at vision and motion, and I'll talk a little bit about some of the new VR technology that has developed out of that work. Some of the other parts of this grant, we had private companies that were developing some new technologies. Post-traumatic epilepsy is a big issue for a large number of folks who've suffered these traumatic brain injuries. And we had a company called Ensight that was developing, much like we were trying to develop a blood test, they were using brain recordings to see if they can pick out very particular signals that could indicate whether an individual is going to go on to develop epilepsy or if they're going to be okay. And so again, another major diagnostic. And then also looking at the neurocognitive effects. VAST was a company with Eric Gusick who basically was using divergent thinking to get a better assessment of some of the cognitive issues and interventions that we could do to maybe lessen those effects of injury. Obviously I'm very biased, but I would say that the overall project over its 2 years was fairly successful. We started 5 new companies that are continuing to develop the technologies that we started. We've had patents filed and awarded. We're up to 7 now, some of which have been licensed and are being further developed in the private sector. We have quite a number of commercial products that are already hitting the market now. We have balance testing, which can provide some incredible insight into which areas of the brain and which circuitry in the brain has been injured. And so that was led by Alex Santos, who actually just recently moved to Kalamazoo, Western Michigan. And then we've created 32 jobs. So these are jobs within the state of Montana, which was the overriding goal of the whole MReady program. And so why TBI? All right, why were we really focusing on TBI? Well, it's definitely, as you guys are aware now, with all the press and the media and the light that's shone on, obviously, concussions in sport, it's a very big public health issue. The numbers that we use to quantify how many people we believe have these injuries, how many occur every year, are a little flawed because the reporting isn't that great. Over the last 5 years, though, as the media spotlight has intensified, maybe even 10 years, our reporting has gotten better. People are keeping better tracks. People are more aware. And so, this number was put out by the CDC and UCSF that they were thinking there was about 2.2 million new cases of traumatic brain injury, or people living with the consequences of traumatic brain injury every year in the United States, and that the annual cost of all the treatments and all the other things that have to happen to help these people is knocking on the door of $77 billion. I'm going to tell you right now that these are both pretty gross underestimations of what that value is. I think the newest numbers are going to put this thing like 120 billion, and it's probably closer to 4 or 5 more recently. Why in Montana? And this was a centerpiece of obviously the MReady. Montana, depending on the stat you look at, and I think this still holds up, ranks 2nd in the nation per capita for TBI injuries. Okay, and we only have a population of roughly a million people, so you can do the quick math. 13% of the population of this state are living with consequences of the traumatic brain injury. So it's 130,000 people in the state. Has a big impact on the economy, healthcare. We also have a very large population of veterans who've served in the 3 most recent conflicts. And while in military case, they come home, obviously have confounding issues such as post-traumatic stress disorder, as well as their concussive and traumatic brain injuries. So it's a very important population here that is living with the long-term consequences of these injuries. And so, this to tackle and help these individuals was the primary goal of our MReady support. What is a head injury? It's going to sound silly to explain this, but in most of the work that I'm going to talk about today is traumatic brain injury that's occurred due to impact. Okay, it's an acceleration deceleration injury. Okay, so you've got a body in motion, the head. It stops suddenly, and then the brain, which is basically floating in a sack of water for all intents and purposes, is able to move, and it contacts at the initial injury. Causes the brain to impact the inside of the skull, and then you also get a recoil injury. So you can actually get damage all over the brain, not just at the point of impact. The mechanisms that we know occur, some of the gross signs, the primary mechanisms that we see, injuries that we see following that, A lot of hemorrhage, a lot of contusion, and lacerations in some cases. One thing that's obviously very common and we know is occurring is cell death. Okay, so the neurons in the brain following these injuries in the regions where the impact occurs tend to die. We were very much interested in these secondary consequences of injury. So when you have the primary injury, it's just the start of a cascade of mechanisms that occur that we believe now over a very long period of time. And that's been, it seems obvious, but it's something that people have focused on more because when people get these injuries and they feel better in 2 or 3 days, they don't get any clinical input or go see a doctor or a PT or anything like that. And what can be happening inside the brain, because it's a closed head injury in most cases, especially in concussion and mild traumatic brain injuries, there's a whole host of secondary mechanisms that are happening, molecular changes in the way that the cells are reacting to the injury, potentially repairing, maybe getting worse. And so, we were really interested in some of these mechanisms, especially the free radical generation and damage as a result of injury to the cells in the brain. Leading causes, this is makes pretty easy sense, really. Falls are the #1 cause of traumatic brain injuries in the United States. There's numbers from the CDC in a report that actually went to Congress. And when falls, quite literally from here to the ground, it's nothing too extravagant, roller coasters or anything like that. It's usually people from a standing position either hitting the ground or as they're going to ground, hitting some object. And that's primary cause for injuries. Obviously, unintentional blunt trauma. A lot of the sports injuries that we're talking about sort of fall into that category. And as that reporting is increasing, this portion of the pie is getting larger also. One positive, I guess, motor vehicle accidents. Obviously a big issue in this state with the size of the state and the amount of travel, the speeds we're allowed to drive, some of the beliefs about whether using a seatbelt or not. In the state of Montana can account for quite a few head injuries. Nationally, though, that number is actually decreasing with the implementation of laws relating to seatbelts and other technologies in vehicles to limit the injuries following a car crash. Pardon the pun here, but I mean this figure basically is just showing at one time it's estimated 1/3 of all deaths due to some kind of injury was due to traumatic brain injury. About 50,000 deaths a year. These numbers again are a little, they're about a decade old now, so that number is probably probably higher. But the real take-home message from this is that it's estimated about 75% of all of these traumatic brain injuries are concussions. Okay, and there's a big debate right now: is a concussion a traumatic brain injury? Are they the same? Is it on the scale of traumatic brain injuries in terms of the physical damage? All I'll say is that as we study this and some of the work I'm going to talk about in terms of the blood-based stuff that we're doing, most of the folks we have involved in the studies have had concussions. They've all had closed head injuries, and they were all believed to be fairly mild in nature. And this is where we sort of want to have our biggest involvement in changing what is being used to assess and then follow these injuries. Depending on the arena, there are a set of tests that clinicians have devised or researchers have devised purely necessitated by need. And so researchers and scientists are going to use any kind of measures they can get to try and quantify these injuries, whether they're accurate or not, or whether they truly pertain to this particular injury. And in these cases, like in a hospital setting or emergency room setting, they actually use a scale that's used to evaluate folks who could potentially be in a coma. This has sort of been the standard here for a while. In the training room, there are written tests, computer-based tests, the SCAT3, the impact. If anybody's doing athletics here or any on the team, you probably do some of this testing before the season starts. I know they do at UM, and then they use that as a baseline. And while there is some value in some of these tests, unfortunately, one of the issues is that they tend to be very subjective. They can be influenced by the person administering the test, because some of the values are based on personal opinion. They are also very prone to retest error or bias from the individual actually trying to take the test. There's lots of situations where if you're looking at an athlete, they will either try and throw the test or influence the test to get a decision at the end whether they want to play or not want to play. And so one of the issues that we're trying to address is that subjectivity. And that's one of the pitfalls. Okay, and so until very recently, a lot of the assessments out there unfortunately fell on this side, on the left side of this figure. And what we hope to do, and many researchers around the world, is to start developing something a little bit more objective. If you had a biological measure, it's much harder to fake a blood test or some other cognitive battery of tests where you have built-in measures to look for if you're trying to throw the test. After these injuries, each one of these injuries is as individual as the person who has suffered the injury. The manifestation of symptomology can be wide-ranging and sometimes very unique to the individual. But one of the broad behavioral measures that we can sort of look at and some of the changes that we notice in these individuals, obviously the most obvious one is cognitive changes in the individual, issues with remembering, issues with concentration. And some of these can go for a short period of time after the injury, maybe a few weeks, and in some individuals they can last many months, even years. Behavioral and emotional. This has become a much more intensely studied arena for TBI because it's becoming well known that certain injuries can cause extreme personality, not even extreme, subtle personality changes where the individual is aware that their behavior is changing, personality changes, which may not always be completely obvious to their peers or family. And that can lead to further emotional stress. Motor changes, coordination, the ability to do everyday things like even walk, reaction times change, sensory, lights, auditory changes following injuries, and then just somatic signs, headaches. One of the studies I'll talk about, every individual that we talked to that suffered, the number one thing that they said, even though they've been medically cleared, is I still get headaches and I think they're from my injury. Okay, so this basically highlights how complex these injuries are, because the organ that's being affected is itself extremely complex. And that sort of just highlight, I use this for some of the STEM programs we do with some of the schools around Missoula. And when I usually ask the kids, hey, what is the brain most like? The number one thing they say is it's a computer. Okay, and it sort of helps because you can use the analogy of a computer chip Okay, and the transistors on the computer chip is a measure of its computing power. Okay, and I think at the time I made this slide, the most powerful chip had 7.1 billion transistors on the wafer. Okay, so if you look at the transistor as a unit, in the brain, the analogous unit for a transistor is the synapse, the connection between two independent neurons, or even neurons impinging on themselves. Okay, and when through the research that's been done, imaging and understanding the ultrastructure of the brain, there's estimates that in a human brain, there's anywhere between 100 to 500 trillion synapses. Okay, so these are the connections between neurons that process information. This picture here is actually from a rat. This is a portion of the brain called the hippocampus, which is very much involved in learning and memory. This was work done at UCS also and San Diego. For a very long time, everybody thought that every neuron in the brain in regions were the same. So all of these hippocampal neurons were thought to be the same. And these guys did this incredible experiment where they tagged all these different proteins with different colored fluorescent proteins. And basically what they would only glow if that protein was expressed in that neuron. And instead of seeing like either all blue for a protein that we know is expressed in that region of the brain, they got this thing called the brainbow. And you've got this incredible diversity in neurons that we thought for the longest time were exactly the same. And so that's another layer of complexity within the system that we're having to deal with. So you can imagine when you injure this, the response of neighboring neurons could be very, very different. The brain changes after TBI. Okay, a lot of the early work and a lot of the continuing work in trying to understand and track traumatic brain injury was done through imaging. Okay, and in some cases it can give us a lot of information, especially on the more severe end of injury. So whatever mild, moderate, and severe are, it's still very subjective. This is an image. This is a diffuse tensor image. So it's basically an image that's made by following molecules of water, aligning molecules of water in a magnetic field. And what it does is it actually helps show where the projections from neurons are forming, because you can track the water inside each individual neuron, and it shows you the networks, the connections that is making. Okay, this is a 25-year-old male, healthy, never had any kind of injuries, and you can see the extensive connectivity. This is a 35-year-old male who'd suffered a number of traumatic brain injuries, concussive injuries in early adulthood. And you can see in this particular case, when you look at that image, that there is a marked loss of connectivity in that brain. And it's not surprising that there would be some deficits or cancer. But amazingly, even in this individual, even with this amount of loss, the symptomologies were extremely subtle. So we know the brain is changing at a structural level. We wanted to study those kinds of changes, okay? So we needed a model that we could use that was reproducible, because as scientists, obviously, we want to try and control as many variables as we can. And so for the project under the MReady, where we were trying to develop these blood-based markers, we needed to limit the variability in our model. And so a collaborator of mine, Thomas Rao, used this instrument. It's called the lateral fluid percussion instrument, and it basically allows us to give a rat very reproducible head injury that mimics a mild. We can actually tune it to be either a severe, moderate, or mild injury, or a concussive injury in the same region of the brain, so it can allow us to study the molecular changes that are occurring in the CNS following those injuries. Yeah, you need to turn the lights down for this one. All right, so this was actually one of the first experiments that we did. Do you want me to get this one? All right. Might get a little dark. All right, so this is an experiment we did to look for a protein that we knew was involved in helping cells recover from injury, and the primary mechanism was mopping up free radicals. Okay, so when a cell is metabolizing and burning fuel and doing all the other biochemical processes it does, nothing is 100% efficient, and you get a lot of side reactions, and you get waste, much like an engine, car engine when you burn fuel. Those waste products can be extremely toxic to the cell, and in normal healthy cells there are a whole slew of mechanisms that can prevent those molecules from asserting any kind of damage to proteins, to cell membranes, to the DNA in those cells. And basically what they do is they mop up the free radicals, and they're a whole slew of antioxidant molecules. The protein I'm talking about here, SystemXC, is actually a transport protein that's involved in supplying a very important amino acid for the generation of an antioxidant called glutathione. Okay, and glutathione is a very powerful antioxidant. And in my graduate work, I studied how this protein functioned, but also the role it could potentially play in things like cancer and other diseases. So I was very interested to see what, if anything, this protein might be doing in traumatic brain injury, as it's such an important mechanism to reduce the potential damage from injuries. And so going into this experiment, I don't mind telling you, I was expecting, based on work we've done in the past, that maybe in the brain what would happen is you'd see an increased expression of this protein to help protect the brain. And what we actually found was the exact opposite in the CNS. So this image here is a brain slice. It's a coronal brain slice at the level of the hippocampus in a rat brain. Okay, so if the nose is sticking out of the screen towards you, and we're approximately halfway through back from the olfactory bulb in a rat across the cortex. Okay, and this is a 7-micron slice. I have a technician, Diane, who's extremely gifted at slicing brains, and she basically took a slice of this brain, and we were able to detect proteins that we're interested in using antibodies that specifically can bind to those proteins. Okay, and then we can put a fluorescent tag on them, and that shows us where this protein is localized in the brain. Okay, and so this protein, SystemXC, that I'm interested in, XCT, is in red. Okay, and you can see that there's a distribution of that protein throughout the cortex. Right here, we're actually at the motor cortex, where we are front to back. We see staining. These are ventricles. These are the fluid-filled holes in your brain, basically, where the cerebral spinal fluid and the nutrients can move in and out into the brain tissue from there. And SystemXC is very highly expressed because it's very important to get cystine into the brain tissue. So if I was to do a zoom up there, it almost looks like a string of pearls. You can see every cell highly expressing this protein. And then places like the striatum, nucleus accumbens, which is involved in addiction, it'll get a little important later. So this is controlled. This green protein is a protein that we're using to mark landmarks in the brain. So this is a protein called GFAP, and we've studied GFAP in brains forever, and it's great because it allows us to know exactly where we are in the brain, because we know its distribution very, very well. And so this is what a normal brain looks like. We did the same experiment at the same time on a rat that had been injured and allowed to recover for 2 days. So 2 days following the injury. So Diane did the same exact thing. She sliced the brain and stained it for System XC. Now, when we first looked at this, poor Diane thought she'd completely messed up an experiment because she didn't see any red at all. And we had hypothesized that it was going to go up. And so she repeated it several times, lots of times. And it turns out that in a 2-day period, using this technique to detect for the protein, we saw an almost complete loss of System XC. So a protein that could be involved in preventing damage in the brain by generating antioxidants was actually gone following the injury. Didn't really make very much sense to us. What we did after that initial finding was like, okay, let's one of these rats recover for a longer period of time. And we went out to 48 days, okay? And she looked at the staining now, and it's hard to see on here, the red's not really coming through, but we actually started noticing that there was a return of the staining. Not very much, but it was starting to come back, okay? One of the other things that was very noticeable was the change in this GFAP, this architectural protein, and how it was changing. Now, in other forms of injury, when neurons are damaged, GFAP is expressed in a support cell called astrocytes. And following injury, these cells can undergo a process called gliosis. Okay? This is a phenomenon that's really studied in spinal cord injuries, because at the site of an injury where the spinal cord is either crushed or severed, what you see is a marked gliosis in that tissue. And what it does is it forms a glial scar, and that prevents the neurons from growing back, so prevents the repair. In this case, you can see that the cells have reacted because there is much more GFAP staining. This staining here actually is where on our LFP machine, we only give an injury to one side of the brain. And this is actually a glial scar as a result of the shock wave that comes from our instrument. So we can actually see where the injury, we can map the injury, and this is very, very consistent in all of our experiments. So it provides a very good internal control. So why is this happening? Okay, and we weren't sure initially. Like I said, we expected it to go up. There was some mechanism occurring that was causing the loss of this protein. Okay. One of the other things that we wanted to check is to make sure that along with those changes, that were the rats producing any of the symptomologies that we see in human patients, okay? And so we did some very simple tests to assess their neurological functions and then their motor functions. And if you look at these two graphs, basically what we're doing here is a neurological severity score. So very much like some of the assessments that would happen if you went into the ER or you went into a clinician's office. They look at they ask you to do a series of tests, and they look at obviously eye motion, all kinds of things, and they give you a score based on how well you perform on these tasks. And in the case of the neurological severity score, the higher the score, the more deficits there are. Okay, and so if we look 48 hours in our rats, if you look at the control rats, you can see they have a very low score. Their functions are okay because they haven't had any injuries. When you look at their injured counterparts, you can see there's a dramatic difference, okay, indicating that there are some severe neurological deficits in these animals that have sustained these injuries. Okay. We were also able to track, much like we did with the staining, are these things improving? Are they staying the same? Are they trending towards healing? And what we found over those 3 time points that we were able to look at, that there is an improvement. Okay, some of the functionality, neurological functionality is coming back. Okay, but again, compared to the uninjured animals at that point, there's still a dramatic and scientifically significant difference. Okay, so even at 48 days, there are some significant deficits. The foot fault score is basically rats are extremely coordinated, okay? And you can put them on a rotating drum and they'll just happily, on the outside, not like a hamster wheel. But if they've suffered some kind of brain injury that's causing motor deficits, what'll happen is you'll see their hind legs slip as they're trying to stay coordinated on the drum. Okay, and every time they slip, we basically count that, and count that as a foot fault. And in a very similar way to the neurological score, you can see that it maps pretty well. And when you look at the control rats, less than 10 foot faults over the time period that we're assessing the animals. But when you look at their injured counterparts, much, much higher. So there's some motor deficits that are occurring as a result of the injury that they've suffered. Okay, and so this all sort of tends to at least give us some indication that in our model we're seeing some of the things that you see in the human population. So looking at those proteins and starting to understand, hey, there's some proteins that are changing over a long period of time that could influence some of the neurological and motor deficits that we're seeing. So we wanted to get a better idea of what kind of proteins could be potentially involved and some of the mechanisms that could be driving some of those deficits. And then obviously, can we use those as measures of outcome for any given rat and then hopefully into humans? Okay, so my technician Fred and I put together this experiment. I mentioned System XC is part of a whole group of proteins that are involved in preventing oxidative stress. So we basically took rats that had been uninjured and injured rats. And for this particular experiment, we started using mild rats because we really wanted to get at these things involved in concussive-like injuries. And we basically did a comparison. We basically took all the RNA from the brain of this particular rat and compared it to one that had been injured. Okay, and we used PCR, quantitative PCR. It's a very common technology. People are developing tests that are used in hospitals and clinics now too, to assess how 84 genes that we thought might be important were being changed in the TBI animals compared to the uninjured animals. And this is basically a heat map. So each square represents one of the genes we were interested in looking at. This bottom row is a whole series of controls. And on this grid, if you see black, that means that that gene wasn't changed in in the TBI compared to the control rat. If there is a protein that's red, that means its expression went at least twofold, I believe. Actually, no, sixfold in the traumatic brain injury rat. Okay, so there's more of that messenger RNA in those rats. One of the things that stood out to us almost immediately was the amount of green on this array. Okay, there seems to be a disproportionate number of genes that are being downregulated. Okay, there's less of those genes being expressed in the injured rat, and that really sort of tweaked our interest because the majority of the genes on this array were actually part of protective mechanisms that are in the brain, in the CNS, to help prevent damage or further damage or promote recovery following injuries. And so that really, why such a large number of these genes would be downregulated so quickly, really got us thinking about, is there an underlying mechanism that potentially is causing this gross change? Now, I have to be honest, I couldn't at this point tell you if these are going down for a reason. Is there a good reason for these proteins to go down? Because we didn't understand the mechanistic value in losing these proteins or not having their activity. Okay, maybe it's a good thing, maybe it's a bad thing, but based on the cognitive and motor outcomes, we would tend to think that it's a bad thing that these proteins are disappearing. So one of the things we tried to do was decipher what could potentially be causing those changes. And I'm going to jump a little bit. So we became very interested in a series of molecules called microRNAs. Okay, and microRNAs are small pieces of nucleic acid, so the same material that makes up DNA, RNA in your body. These are very small chunks, anywhere between 8 and 22 nucleotides. So that's the number of bases that comprise those microRNAs. Okay, and what these microRNAs can do is they can actually very powerfully regulate protein expression. So in any given cell, you've got the DNA, which is basically like the database of instructions. When you want to make a protein, the RNA gets transcribed off whatever portion of the DNA codes for that particular gene. And in normal circumstances, that RNA is then translated to give you the protein that the gene codes for. Okay. What microRNAs can do is they can intercede at the point of translation. Okay. So once this specific instruction has been generated off the DNA, as it's going to the ribosome, you can have these microRNA molecules, really powerful regulatory molecules, which actually can directly bind with the RNA in a very specific spot at the tail end of messenger RNA and prevent the protein from being synthesized. Okay? And in some cases, these microRNAs can actually lead to the direct degradation of the message itself. And in other cases, it basically gums up the work in the ribosome, the organelle the unit responsible for translating the message into actual protein. Basically, the RNAs get up in there and just gum up the machinery and prevent the protein from being made. And then this is sort of more of a schematic. I don't know how clear that is there. If this is the part of the messenger RNA that codes for the protein, this is the 3' UTR, the 3' untranslated region at the tail end. And this shows a microRNA binding to its target site. And again, these are very, very powerful regulators, as we found out. They were discovered about 10 years ago, 10, 15 years ago. And a lot of the work, the original work looking at these microRNAs, like a lot of new mechanisms, was actually sort of in cancer and the potential use of these as diagnostics for cancer. We basically came in and we think we can use these things as potential diagnostics for traumatic brain injury. And so we did basically a thought experiment in a computer. And so, as I mentioned, microRNAs can target, a single microRNA can actually target hundreds of proteins, hundreds of genes in humans. But it's all dependent on specific binding and finding recognition sequences. So what we did was we looked at the proteins that we knew that were changing and tried to see if there was microRNAs that were common between all those proteins. So we basically threw all this into a computer and had it spit out, tell us what microRNAs could potentially be responsible for this. Okay, so we used free software actually to basically look for target sites for these microRNAs. And based on that study, we were basically very much like the gene array I just showed you. We created about 84 microRNAs and said, let's see how these ones are changing in the uninjured rat compared to the injured rat. And what we found, and so this graph is showing some of that data. Okay, so on this graph, you've got this solid black line, okay? And all of the individual black dots, all of these dots are a unique microRNA, okay? And if one of those dots sits along that central line, that means there was no change between the control animal and the expression in the injured animal, okay? This dotted line above it indicates a microRNA that was expressed at least twofold higher in the injured animal, okay? So you can see these red dots, I don't know how clear that is. There are some of these microRNAs that were highly expressed in these injured animals. Okay. Surprisingly, there was only one that we found that was actually downregulated. So we obviously started to focus on these particular microRNAs, and this is how you name these names. There's, as of I think this summer, there are now 2,000 microRNAs that have been discovered in the human genome. That number is definitely going to increase because these molecules reside in what was thought to be junk DNA. After the Human Genome Project, where we figured out there was about 20,000 genes in the human genome in all the bases that comprise, and we thought there was lots of areas of non-coding junk. Turns out a lot of these things in other RNA species, long non-coding RNAs, actually reside in those genes. So it's definitely, in my mind, moving from a DNA world to an RNA world. So one of the things we wanted to do was, okay, we've identified these microRNAs. Do they have any effect? The computer told us that they should do something, okay? And that's one of the issues when you're working with genomic data, okay? It's all based on prediction. It's a computer telling you, yes, I think this will interact with this particular gene, right? You still have to go back and do the experimentation, because what you find is a lot of the predictions that the computer makes is purely mathematical. It's not based on any solid science, biological science. And so we had to go back and confirm, just because that these microRNAs could actually affect the expression of this one protein that we were interested in. And so this work was actually done by a high school student that worked in my lab for a year. And she basically grew cells that I knew could express high levels of the protein that we're interested in. And then she was able to transfect. She used a chemical reagent that could take RNA for these microRNAs that we were interested in and get them into the cell. And then she could quantify how the protein expression in those cells was changing. Okay, and so to go from here to here, there's a lot of steps that I'm not showing here. But she very diligently characterized these microRNAs that we were interested in, and to our relief, showed that these microRNAs that we had predicted could interfere with the protein expression of these, the protein expression of System XC actually did. Not all of them, and to various degrees. So on this graph, these are cells where we took dummy RNA and we transfected into the cells. It shouldn't have any activity. And we used that as our control. And when we looked at the XC expression by Western, we made that 100%. So this is the normal amount of expression. And then we could take the cells that we had treated with the microRNAs, she had treated, and look at how much there is relative to that control. And you can see after a 2-day exposure that some of these microRNAs could cause a fairly significant drop in the amount of protein that's being expressed in the cell. So that was one piece of evidence that sort of suggested that, yeah, these microRNAs could cause the effects that we were potentially seeing in the injury model. So at this point, and I was talking to Beverly about this was one of the points where, as a research track faculty, when money starts getting tight and you're like wondering when the next paycheck is coming from, we were actually brainstorming how we could do cheaper experiments, because animal experiments are really expensive. Okay, and that was when we sort of like, well, hold on. If we believe that these microRNAs are changing in the brain, and I had done an experiment to show that some of these microRNAs in the injured rats, in the blood of these animals, these microRNAs were appearing too and being elevated that matched the injury animals. Can we do this in humans? Humans, way cheaper to work with. And so we got permission from the athletic department at the University of Montana and the coaches of the Grizzly football team to basically bleed some of their players and see if we could detect any of these microRNAs in these individuals, okay? And so we basically enrolled 49 of students, all years. The mean age was about 20 for that group. We also included in our study a set of non-athletic controls, so folks who weren't athletes, so that were age matched, so primarily students that hadn't participated in any kind of contact sport for previous 3 years. When we spoke with the football players, we asked them to be very honest, and it was all based on self-reporting. Okay, we didn't actually have any access to medical records, so we wanted their players' input on whether they believe they've had a concussion or not, if they've had a medically reported concussion, if they've sustained one during practice or play. The coaches were totally okay with them disclosing that information under our IRB. And so we put together a group of about 28 players that claimed that they, in the last 3 years, had not had an injury. And then we had a concussion group of about 21 players. And one of the things that we actually did was, if they said they had a concussion, we also asked them, do you have any kind of symptomology that you believe is potentially linked to your injury? So whenever you got your injury, do you think, concentration, headaches, all of that kind of stuff? And what we were able to do is almost able to split them in half. Okay, there was a group that said, yeah, no, I'm fine. I haven't had any other issues since I've had my 1 or 2 concussions. And then we also had a group that, even though they'd been medically cleared by the trainers and their doctors, still were reporting some kind of symptomology. And then a really interesting observation that we made was that the average mean time for these individuals in this study who'd had injuries was about 12 months, so a year out. Okay. So much like with our rats, we wanted to get some kind of assessment of cognition, see if there are any cognitive deficits in these individuals. And this was very early on. And of the group that reported symptomologies, this is a breakdown of all the symptomologies. And I said at the top of the talk that 100% of the guys with some kind of symptomology said they had headaches, and that headaches were a constant issue following their injuries. Another thing that they all reported are memory issues, some kind of memory issue. Okay, forgetfulness, inability to remember stuff in the past, kind of thing. And that varied, but we sort of grouped that together. And then the other one that everyone reported was concentration. They had a hard time concentrating on tasks. And you think about this, these are football players that have to learn plays, they have to learn routes, all these kinds of things, as well as go to school. Right? These are all student athletes. And so these 3 factors alone could have a big impact on their success in both their academic and their athletic endeavors. And then a whole series of other insomnia, mood changes, balance issues, like I said, probably related to motor issues, depression, mood affect changes, in a third of the individuals, but nobody reported any kind of vertigo. Okay, so to do the rat testing in humans, does everyone know what assignment is? Who's done assignment before? Not that many. Right, so this is a game when I was a kid. It's basically sequence building, right? It plays a tone and a color, and you have to replicate the sequence. And so basically what we ask these students to do, these athletes to do, is go as long as you can. When you make your mistake, we're going to have you do another one. When you make a mistake, we'll have you do another one. And that was an important thing here. When we did this Simon testing, what we noticed, and there's a lot of literature on this, when you test folks who've had potential head injuries, and you're using certain measures, okay, a lot of the time when you do one trial of a test, what you actually find is that the TBI individuals actually do okay. They're not that different from the control patients or the control subjects. Okay. What my colleague Tom devised, because he'd seen a similar effect in the rats, was let's load these individuals. Let's not give them a break. Let's make them do it again. And what we found was when you do that, when you look at the controls, which are in white, okay, you can see those individuals in 3 trials stay very, very constant. Okay. But in folks who've had injuries, we see this drop. Okay, that's significant. Okay. We did another test after we did this one to back that up. It's called a recall test. This is a very classic old school test. Okay, a bunch of words, different fonts, different sizes on a page. You get a certain period of time to look at it. And then we take it away, and then you try and recall as many of the words that you saw on that list. Okay. And when we did that in controls, you can see with each trial, there's an improvement. Okay. There's a similar, or a non-significant improvement in the TBI patients, but they are always lagging behind their uninjured counterparts. And this is in the football players. Okay. And so in these players, even though they've been medically cleared, okay, and again, this is a very simple test, it's not very involved, we could pick up differences. And, you know, this was 3 years ago, 4 years ago. We've now, as part of the MReady, one of the things that we've developed is a virtual reality setting for this kind of testing that is unbelievably more in depth, obviously, than the simple Simon test. But it gives us very much similar data. We can do a much larger, broad breadth of measures with that testing using the VR. And this actually also, we find, relaxes the patient. It's more like a game. And so it puts the subjects more at ease. Also allows us to see if someone's trying to throw or manipulate the testing, because we have very precise baselines. We know how fast eyes can move. We know how fast normal auditory visual integration occurs. These are numbers that we know. And so if someone is trying to throw the test, they'll be very abnormal. And we can also track the eyes. So this is one of the technologies that was developed from that work. Getting back to the microRNAs, and sort of to wrap it up, when we did that testing, I also took blood samples from all the individuals, okay? And we looked for the microRNAs that we had predicted in our animal studies. And this is just 3 of them here, okay? And so in each one of these graphs, you've got the non-athletic controls, so the normal folks, the football players that reported no concussions, football players who had a concussion but don't have any long-term symptomology, okay? And then the football players that, even though they've been medically cleared, continue to report some kind of symptomology, okay? And in these experiments, basically what we did was we took the level of the microRNAs in the control individuals and saw how many fold different the expression was in the plasma of the other groups, okay? So everything's relative to control. So if this is 1, in the case of MIR 142, okay, when we looked in the plasma of those individuals, there was 5 times as much MIR 142 floating around in their blood, okay? Similarly, and this is a slightly different scale, when you look at 432, again, very little difference actually between the normals and the football players without concussions. But you see an increase in the concussion group, but a resoundingly more significant increase in the individuals that continue to have some kind of symptomology. And with 223, that was even more apparent. It seemed to almost select for folks who'd had the injuries and we're having symptomologies. So we've basically taken this and tried to do a larger assessment. We're in the process of setting up some blinded studies where the outcomes, we're going to obtain blood samples from folks who've had injuries, but their outcomes are known. We'll be blinded to those, and we're going to see if our microRNAs can distinguish and identify the patients correctly. The ones that can this test show that these guys had a concussion, and two, are potentially having long-term symptomology. So it's a retrospective study. So we're trying to raise funding right now to do this in a much larger cohort. One of the other things, one of the things I didn't mention, to do this work, I have to take a blood sample. I have to process that blood to get the plasma, and then I have to isolate all the RNA. Then I have to do a quantitative PCR experiment, and then we have to do an analysis. Any given week, that takes, with all the coordination we have to do, easily a week, if not more, to get a number that we can use to do the comparisons. Okay, so one of the things as part of the MReady that we're also doing is developing a new technique, a new methodology, where we can get an answer in 30 minutes. Okay, so quantitative PCR requires all kinds of enzymes, lots of temperature-dependent steps, chemicals, an instrument that can cost anywhere up to $50,000 to $75,000. Okay, what we're doing, and we're actually working in collaboration with researchers at MSU here in the state to develop an isothermal technique. So, I mean, the chemists in here probably understand this better than I do. Where we can basically, they've made a detection system that can detect the presence of these microRNAs in a raw blood sample and then produce a fluorescent signal to say yes or no right now, like a digital readout that it is present. And that's important because some of these microRNAs, when we did the initial studies, in normal patients, it was at the very lowest level of detection for these microRNAs. So in a normal, uninjured adult, the expression of the microRNAs we're interested in looking at for traumatic brain injury are actually not there. So you're going from nothing to something. And so we're trying to develop a digital test that will basically, it's like a switch in the presence of microRNAs in the blood from no signal to signal. And so this is how big it is right now, but we're hoping to get it down to an instrument the size of a diabetic glucometer. I know I'm running a few seconds long, so this basically just summarizes everything I've talked about as we try and understand the molecular effects and that of traumatic brain injury and how we could potentially use that knowledge to develop more accurate, objective measures to better understand and track these disorders. And then final slide, I have to, obviously didn't do any of this by myself. My partner in crime, Dr. Thomas Rao, who developed the animal model at UM and helped me get on the way to discovering these microRNAs, Diane Brooks, and did a lot of the staining that you saw, a lot of the Western analysis that I didn't show. Fred did a lot of the molecular biology, the gene arrays. Eric helped Tom with the animal model. Once you start that project, you have to watch the animals every single day. It's a thankless task. Night and day, there is no 9 to 5. So he gave a lot of time. Paige Fletcher was a graduate student who rotated through my lab, and Carolyn was the the high school student from Sentinel High School who did phenomenal work. She had great hands. Funding resources and support at the university and the university system, the Neural Energy Center, Center for Structural Functional Neurosciences, in the School of Pharmacy, in the College of Health Professions and Biomedical Sciences, I don't think it exists right now, but the Neuroscience Institute at St. Patrick's Hospital was an early adopter of helping us get these projects started. And then obviously the research office at Main Hall. And then these are all the funding agencies that have been very kind to us supporting this work. To all the research scientists out there, I'll say this whole project, everything that you've seen here, started with a $5,000 grant that I got from the research office at the U of M. And so without that, none of this would have been possible, and just to show that it can springboard you into a successful timeline to acquire funding and keep the research going. And with that, I'll say thank you and take any questions you may have.